Greetings to you, lovely students. I welcome you to today's class. Today's class, I'll be taking the topic plant and animal cell, their structure and functions of the cell organelles. Cell is a very important part of living organisms. All living organisms are made up of cell, and that is why cell is regarded as a basic functional and fundamental unit of all living organisms. An organism can either exist as unicellular or multicellular. So, no cell can exist on its own. That is, what I'm trying to say is that a cell is not just formed by itself. A cell is usually formed from another cell. So the studying of this cell is actually very important, and the study of cell is actually referred to as cytology, and the study of group of cell, which is the tissue, is referred to as histology. Now, in today's topic, I'll be taking you the objectives, definition, and classification of living organisms based on number of cells. We're also going to look at the scientists who were involved in the discovery of cell. We'll also be looking at the cell theory postulation. There are about three of them. We'll be looking at parts of a microscope. There are about two types of microscope we'll be looking at. We'll also be looking at the structure and function of cell components such as the mitochondria, the lysosome, and the rest. Now, definition and classification of living organisms based on number of cells present. We have about two types of cells, but first, what are cells? Cells can be defined as a basic and structural unit of all living organisms. That is, if you look at an organism, the basic part of it and the fundamental part of it is referred to as a cell. So a cell can either exist as an organism can either exist as a unicellular organism or multicellular organism. Unicellular organism, they possess just one type. They're made up of one cell. Examples of such is the bacteria, paramecium, amoeba, and the likes. Then examples of multicellular organism, we have the hydra, we have volvox, we have humans, we have cats, we have earthworms. All those ones are made up of several cells coming together to form an organism. Now we look at the discovery of the cell. A structure that looked like a cell was first discovered by Robert Hooke in 1665, and so that's why Robert Hooke is regarded as the father of cells. And this is the microscope that was first invented by Robert Hooke. Notice all the parts. It has an oil reservoir, specimen where you put the specimen, the water flask, the oil lamp. Now this is actually a crude microscope, but it did a lot of work in helping to discover cells as we have today. Now the work of Robert Hooke was further done properly by other scientists such as Dujardin, who was involved in the proposal that cells discovered by Robert Hooke contained living semi-fluid. And we also have the Antonio van Leeuwenhoek, who invented a powerful microscope and examined a drop of pond water. He also observed the human blood cells. Then we have the Haeckel. Haeckel was postulated that nucleus was responsible for storing and transmitting hereditary characters from one generation to another. We also have Matthias Ledin, who postulated that all plants comprised of cells. Now, the cell theory postulation, we have about three of them. It says that all living organisms are made up of cells. Another one says all cells arise from pre-existing cells. And the third one is that cell is fundamental unit structure and functions of all living organism. Now, the compound microscope, we have about two types of instruments that are used for studying cells. The first one is compound microscope, and the second one is the electron microscope. Now, the compound microscope is much more simpler and is widely used in laboratory today compared to electron microscope. Electron microscope can only be found in few places because of the way it is made up of. Now the compound microscope has about 10 parts. You have the base, we have the mirror, the stage clip, the condenser, the handle, the eyepiece lens, coarse adjustable knob, and then the last one, which is the fine adjustment knob. Now the base is a metallic stand and a solid part of the microscope which enables it to rest properly. While the mirror, as you can see in the picture here, the mirror is a plain mirror which helps to direct light to an object so that it can be seen properly. Then the next one is the stage and the stage clip. The stage is a square place where a microscopic slide is to be placed for observation. Then we have the eyepiece lens, which is the part which the observer places his eyes for viewing the object. Then the coarse adjustment knob it helps to move the stage up and down to bring the specimen to focus. And the next one is the electron microscope, which is a much more powerful type of microscope that was invented by Knoll and Ruska in Germany in 1932. Now, the electron microscope can magnify an object up to 2 million times, and for that reason, electron microscope is not for regular use by laboratory students or lab attendants. The electron microscope uses signals arising from the interaction of an electron beam with a sample to obtain information about the structure, the morphology, and composition of a sample of a sample. Now, the electron microscope is not an ordinary kind of microscope because it gives about 2 million times the magnification that can be given by the compound microscope. The electron microscope uses beam of electrons to get the greater resolution power. Now, under the electron microscope, the photoelectron micrograph of a chloroplast can be seen. Now, this next one is the structure and functions of cell components. The cell is made up of about 3 major parts: the plasma membrane, the nucleus, and the cytoplasm. Now, the nucleus and cytoplasm is what makes up what is referred to as a protoplasm. Okay, now the protoplasm has a lot of other cell organelles in it, but the cytoplasm is just like an empty space in which it's like a fluid in which the organelles float on in the cell. The cell is not actually flat, it's usually either rectangular or spherical. Spherical in animals, then it's cuboidal in plant cell, and the animal cell is different from the plant cell. As you can see in the picture, we have the plant cell has a definite shape while the animal cell does not have definite shape. Now, as you can see in the picture here, we have the plasma membrane, which is a thin and flexible covering that encloses living protoplasm of the cell. The plasma membrane is selectively permeable in nature. It can only allow certain molecules to pass through it. It's not all molecules that can pass through the plasma membrane. And then we have the nucleus. As you can see, the nucleus has the nuclear membrane, the nucleolus, and the chromatin. The nucleus is the most conspicuous and important part of the cell because it is responsible for the replication of the cell. The nucleus carries the hereditary information of a cell and also an organism. Then also we have the other cellular components like the endoplasmic reticulum, which is essentially an interconnected network of tubules, cisternae, and vesicles. The endoplasmic reticulum is responsible for the transportation of proteins, lipids within the cytoplasm. And then we have the mitochondrion, which is popularly referred to as the powerhouse of the cell because it is involved in the generation of energy. Now, the plant cell is different from the animal cell in this regard. The cell wall of the plant cell is usually rigid. It has a large central vacuole. The chloroplast is present in plant cell, whereas it is absent in animal cell. The vacuole is usually large in plant cell, whereas it is small in animal cell. The lysosomes are usually rare in plant cell, but it is actually very common in animal cell. The lysosome is also referred to as the suicide bags in animal, because it is involved in the removal of at least it can the cell can destroy itself using the lysosomes.